Welcome to this class on Neuroscience of Human Movement. So, today we will be talking about spinal cord injuries and CPGs, central pattern generators. So, we have been talking about reflexes, monosynaptic, oligosynaptic, polysynaptic, pre-programmed reactions, etc. Many of these are thought to be spinal processes, and that means the movements that are generated as a result of reflexes or the processes are probably involved in the spinal cord. Of course, questions remain about the pre-programmed reactions. So, many of these are spinal processes. Suppose something goes wrong in the spinal cord or there is some injury, what are the various things that could get affected? That is the question. So, in today's class we will be talking about a particular form of a problem called spasticity, which is actually another version or a particular form of hyperreflexia, a case in which the reflex responses are greatly exaggerated. Okay. And then we will talk about motor neurons or groups of motor neurons that generate patterns of rhythmic activities present in this spinal cord, especially in lower animals, especially in non-human species called central pattern generators. Why non-humans? Why not humans? We will say later. Okay. So, problems of abnormal muscle tone. There are so the muscle tone can either increase due to a problem, due to an injury, or it can decrease due to an injury. So, if there is an abnormal increase in the muscle tone, so what is muscle tone? Let us remind ourselves of what this is. Muscles are always at some tension, they are always in some non-zero tension throughout, especially when the person is alive and active, right. When the person is in coma, when the person is sleeping, this is different, but in an awake person there is always an amount of non-zero activity which is called as baseline activity. This is what is measured as baseline EMG activity, right. So, this is also called as tone or some residual tension that is present in the muscle even when the muscle is not active. So, this is true of many muscles, almost all muscles. Okay. When an injury happens in the spinal cord or in the upper motor neuronal cases, what happens is that tone is either greatly exaggerated or greatly reduced, or in other words there can be complete flaccidity. Flaccidity means looseness, the muscle is no longer tight, there is no more tension. This case is the case of abnormal decrease of muscle tone called as hypotonus or hypotonia. In some cases also called as hypo means reduced, tonia reduction in the muscle tone, is it not? Hypotonia. In many cases what also happens is an abnormal increase in the reflexes. So, for the same stimulus the response will be greatly exaggerated. So, if I compare before and after injury, right, before injury if the response was x, the response after the injury can be many times x, right, 3x, 4x, 5x, whatever. This is also called as hyper. Hyper means an increase, tonia means increase in the muscle tone. So, okay. Now, a particular form of hypertonia is spasticity. In this case rigidity is dependent on the rate at which the stimulus is applied. So, this is called as rate dependent rigidity. So, there is general rigidity or general hypertonia and there is spasticity. Spasticity is not hypertonia. So, or in other words the general hypertonia refers to only the change in the level or so if there is a if the change in the stimulus is at one level, then the response is something. But if I apply the same change in the stimulus at a faster rate, right, then also the response does not change. In that case you have some form of general hypertonia, or in other words the rate does not affect, the rate has no effect. Okay. So, in this case whether I apply the stimulus fast or slow does not matter, however I apply the stimulus the response is the same. In spasticity it is special, the same amount of stimulus if I apply fast the rigidity is going to be great, the same amount of stimulus if I apply slow or really slow the rigidity is going to be small, or in other words. So, suppose I had a case where I am slowly increasing the rigidity like that, stimulus that I am applying, this is the displacement axis say for example. Okay. So much is the displacement. If the same displacement is achieved in a shorter time, right. So, that means the rate has increased. So, I am pushing very fast. When I am doing something like that the rigidity is greatly felt in spasticity when compared with this. Let us remember in both cases the displacement is the same, the amount of stretching that is applied is the same, but the response depends on the rate. Okay. So, in other words this is dependent on the rate, right. So, this means this is a phasic phenomenon. So, this is more like so we discussed phasic stretch reflex and tonic stretch reflex. We also discussed the case of the 1A afferent where the response is due to the rate of change of the length, is it not. So, or in other words the velocity. So, if I do something fast the response is going to be great, or the reflex is going to be great, or the rigidity is going to be great. The same stimulus if I apply slow the rigidity is going to be lower. If I do it really slowly there is going to be very little or no rigidity that is going to be felt. This is the special case of spasticity. Why does this happen? What is the pathophysiology of spasticity? It turns out that spasticity is present in many cases of motor neuronal diseases, not just one. The pathophysiology of spasticity is very poorly understood. We do not know why this happens. At least it is not clear for us as to what are the neurophysiological mechanisms above the level of spinal cord that facilitate or that could control the development of spasticity. So, but at least what is known is that we could control or manage spasticity amount by the application of specific drugs to the 1A afferent system. So, in other words it is widely believed, the hypothesis is that this is due to some differences in the way that the 1A system behaves. So, if we could mimic the presynaptic inhibition in the 1A system, right, which we have seen in the previous classes, if I could somehow mimic the presynaptic inhibition, then I have at least found one way to manage this disease. This is what is done in the nowadays. So, there is a particular drug called baclofen which is applied intrathecally to mimic or to induce presynaptic inhibition in the 1A system, thus reducing the reflex responses, right. Of course, this cannot this is not a complete cure for the problem, but at least it is some solution. Okay. So, this is spasticity, and there are other forms of hypertonia that are not discussed. Another point is that with the difference between hypertonia and hypotonia. Now, it is in general one thing is known that if you find hypertonia, there is an increase in the muscle tone or increase in the reflexes, right, or this is also called this is usually accompanied by another called hyperreflexia. Okay. Usually these two are interchangeably used, they are different although. So, when these things are there, the problem is central, the problem or the lesion or the injury is either in the brain or in the spinal cord. Okay. Hypotonia on the other hand, or decrease in the muscle tone, or hyporeflexia or flaccidity happens usually due to a damage to the peripheral system. What is the difference? So, the peripheral system means the motor neuronal connection from the spinal cord to the muscle, and the afferents from the muscle to the spinal cord. Okay. This forms the peripheral system. If the damage is there, if the damage is on the connection from the muscle to the spinal cord, so that is the peripheral nerve, right. So, if that is damaged, you usually have hyporeflexia, okay, or hypotonia. If it is in the spinal cord or in the brain, there is hypertonia. So, which means that depending on what the symptom is, depending on whether the symptom is hyper or hypo, we can get some idea of what the lesion area could be, where the problem is approximately. So, this is some information about the location of the lesion, location of the injury. Okay. So, what happens with spinal cord injuries? What happens is that if spinal cord is transected, right, depending on the level of transection, depending on where the spinal cord is transected means it is cut, right, at a particular point, depending on where the damage will be felt on parts below that level usually. But what immediately happens after the transection of the spinal cord is that there is very little, or in some cases no reflexes that are found for the initial period immediately following the spinal cord injury. So, in other words, there will be no response to stimuli that is applied. So, that is a spinal cord injury, a stimulus is applied, no response, right. So, or very little, or a smaller amplitude response or very little response. So, this period during which reflexes are reduced or sometimes even reflexes disappear is called as spinal shock. Okay. And then after spinal shock, what happens is after some time during recovery, which takes time, after several weeks in humans, several months in humans actually, there is recovery. Slowly what happens is the reflex return, but when they return, they are back with a bang actually. What happens is that the reflexes are heightened. For the same stimulus, the response will be much greater, or in other words greatly exaggerated reflexes, or hyperreflexia is what you find. So, and it turns out that the time of recovery between the spinal cord injury and the state of hyperreflexia changes between species. Okay. So, let us see, we are interested in humans and human rehabilitation mostly, is it not? So, in humans it can be weeks or even months in some cases, okay. Sometimes no recovery. In non-human primates, macaques, bonobos, whatever. So, it can be days, max of a week. In non-primate mammals, other mammals, that is cats, dogs, etc., right, hours to days, maximum 1 day, right. In rats and mice, other smaller mammals, small mammals, right, hours, half an hour, 45 minutes. So, something must be special, something must be different. So, as you move up this from a lower mammal to a higher mammal, the time it takes for recovery increases, right. this should give us an idea of the nature of rehab that you could expect depending on the injury. So, so also that means that that the amount of supraspinal influence on the reflexes is great as you move from the bottom to the top. As you go from here to here, the influence of the supraspinal centers in the control of reflexes is greater. In other words, in humans there is very fine control or great control of the reflexes is exerted by the brain and higher centers. So, when that control is lost, right, what you see is recovery takes more time. In the other animals, there is less and less control as you go down from the humans to the from to non-human primates to big mammals to small mammals. As you move down, what you find is that the spinal cord has more and more autonomy. So, that means the possibility of recovery and the time it takes to recover is better. So, but what causes spinal shock? Spinal shock is believed to be caused due to the removal of the facilitatory mechanisms from the supraspinal centers. So, or in other words, I remove the facilitatory mechanisms that come from the brain. When I do that, sudden loss of this is felt as spinal shock is. So, there is not a response, and that is what we call as spinal shock, but what causes recovery itself is poorly understood. Why, how recovery happens, what causes recovery or what could facilitate recovery are not well understood. If we can understand that, we have solved a great problem. So, we have not understood what causes or at least to some extent we have understood the process of recovery. More understanding of the process of recovery is required at this stage. Now, what else can facilitate recovery? Again, little understanding, not clear understanding. Okay. So, another thing is also to note, this will also be discussed in future classes, especially the case of stroke and upper motor neuronal syndrome and lower motor neuronal syndrome. When we compare in future classes, we will discuss, but I will just mention here is the importance of recovery or the differences in recovery between lower animals and higher animals, especially between humans and other animals. In humans, what you see are unique projections to the distal forelimbs or hands and fingers get unique projections from the alpha to the alpha motor neuronal neurons from the upper motor neurons. So, from the motor cortex there is monosynaptic projections to the alpha motor neurons that control hand and fingers in humans. This is a uniquely human feature that is not seen in other animals. If this is compromised for whatever reason, you have a spastic or hyperreflexive human, they are not, the chance of recovery of finger function or the distal most upper limb function is compromised to a great extent, and you are probably going to have very little or no recovery. So, but with what is the cost of having, so that is probably the cost of having a fine dexterous instrument, right, that is the hand. When compared with other animals, when compared with other mammals, when compared even with monkeys, right, non-human primates, the dexterity in the hand is, in the human hand is much greater, right, and that is because we have supraspinal control, direct projections to the fingers, right, not to the fingers, actually to the motor neurons controlling the fingers, right, but that is not there in monkeys or in other animals. But that also increases the probability that they will recover better in case they have an injury when compared with us. So, probably, right. So, this will be discussed in greater detail in future classes. So, this case is the case of hyperreflexia. So, remember we were studying reflexes, we asked why do we study these things, what is the point. I said that by studying this we will get an idea of the presence or absence of disease, right. So, a simple clinical examination can tell the clinician as to what could be the possible location of the lesion, no imaging needed. Well, at least the first line of diagnosis, at least at the first line of diagnosis, the clinician can get a rough idea of what could be the possible location of the lesion approximate. So, it is not going to be perfect. Of course, imaging, with the help of imaging you can get a better idea, but at least just with a simple clinical examination you get some idea. Okay. So, that is what is used in the, for example, the knee jerk the tendon tap experiment, right. We tap the tendon and there is going to be a response, or the T reflex, right. This is going to be heightened in after a spinal injury. So, that means this person is in the state of hyperreflexia. So, you know what is going on, or there is no response. So, you know the person is either in spinal shock, or the lesion is only at the level, at the peripheral level, not at the central level, one of these things. How do I know that the lesion, how to confirm that the lesion is at the peripheral level, not at the central level? Well, simple, right. So, what I could do is I could test this on other muscles, especially muscles at a different level of the spinal cord, right, at a higher level if I test, and if this is not seen or normal reflexes are seen, that means this is probably a peripheral problem, right. If it is a central problem, either the central problem is at a higher level than both the test sites, or it is in between these two, is it not. So, that is the case of hyperreflexia. Okay. Now, what is present for walking, rhythmic activity in the lower animals, right. In the lower animals, rhythmic activities such as walking, swimming, feeding, or flying, insect flight for example. So, there are birds, small birds and insects that flap their wing at multiple times, several hundreds of times a minute for in some cases, right. So, that must happen using an oscillatory mechanism, rhythmic pattern generation, right. And even in humans, for example, respiration is not always consciously controlled, right. Even when you are not consciously controlling, respiration happens. So, that means that there must be a rhythmic pattern in which you know this must happen, or there must be a rate at which it should happen, right. So, how are these things controlled? Well, these things are believed to be controlled by a group of neurons or a neuronal network in the spinal cord. So, central nervous system in probably the spinal cord. So, which is also the reason they are sometimes called as the spinal pattern generators, because they are present in the spinal cord. They are sometimes also called as the spinal pattern generators, but mostly they are called as a central pattern generators, or CPGs. So, this is a neural network that is capable of generating an oscillatory pattern of motor activity without you know inputs from receptors. So, that means there is going to be spontaneous activity. So, by the way, spontaneous activity for breathing is good. By the way, spontaneous activity for breathing is a good thing, we need that, because regardless of whether I am awake or not, I need to continue to breathe. So, that is a good thing. However, for walking, spontaneous activity is not needed, or in other words, I want to have spontaneous activity when I want and inhibit that spontaneous activity when I do not want. I do not want to keep walking all my life, right, all the time. So, there needs to be control. This is true even in lower animals, right. It is not like that the insect is flying all the time, it is not like the birds are flying all the time, it is not like they are swimming all the time. So, there is a need for us to stay stationary when we want and to move when we want, right. So, that is not true of respiration though. So, respiration is different. So, there is a need to control that, there is a need to inhibit this. So, these rhythmic patterns or this generated rhythmic patterns by the CPGs depends on actually three things. First is properties of individual neurons. So, within that network it could have many neurons. So, individual neuronal property or the cellular property is one. So, that and properties of synaptic connections between these neurons. So, and how they are interconnected with each other, right, the pattern of interconnection between neurons. And importantly, the following is has been observed, that in specific cases, right, there is what is called as post inhibitory rebound. What is this? In say a set of neurons that excite or inhibit each other, as soon as an inhibition, a source of inhibition is removed, the probability that that particular group or that particular neuron will get excited is greatly increased, or there is a rebound in the excitability of this, a brief increase in the excitability of a neuron following an inhibitory period, following the removal of an inhibition. So, an inhibition was there, when I remove the inhibition, the excitation is greatly increased, for a brief amount of time. In that time, if we excite, right, there is going to be an excitation, right. So, if there are two groups of neurons that excite and that inhibit each other, right, then if for some reason one of them is spontaneously excited, there is going to be mutual excitation and inhibition followed by one another, or some sort of an oscillatory output can be expected. So, mutual excitation or mutual inhibition is a critical crucial mechanism for establishing this sort of oscillatory outputs or rhythmic output in a group of neurons. So, how does this happen? Importantly, how fast you want to move is a decision that is made from especially we like to think that it is a decision that is made in the in the higher centers such as cortex, and it reaches midbrain locomotor regions. We are going to see what is the critical role of this in future classes, right. So, there is a region in the midbrain that is responsible for controlling locomotion, walking behavior, right. This is called as a CPG timer. I will refrain from using words such as that, but this is somehow encoded in the central pattern generator to generate to the train of pulses that you want, right. So, to form the pattern that you want and the amplitude that you want, but it is not just that, right, there is more than that. What is also present is, so ultimately what you want to do, ultimately you are interested in exciting the locomotor alpha motor neurons, or alpha motor neurons that are responsible for walking, right. So, they must be excited, but not all simultaneously. If all of them are simultaneously activated, there is going to be just co-contraction. So, they will all have to be activated rhythmically. This is what you want. So, extensors and flexors, so this is extensors, these are flexors, are excited one after the other by the spinal motor neuronal pools, and what also happens is the force is produced. So, that means when a force is produced in one Golgi tendon organ responsible for that muscle, and the muscle spindles of the antagonist muscles start firing and reflexes take over from that point. That is, these are these, right. So, these are stretch reflexes or reflexes in general, information coming from 1A afferent, which is muscle spindle, 2 afferent, which is muscle spindle, and 1B afferent, which is a Golgi tendon organ. All this information and stretch reflexes also serve as input to the actual output that could come. And what is more is, there is of course intrinsic muscle properties or the passive elasticity of the muscle that will initially resist movement, and after that there is going to be resistance that is offered from the reflexes. We have seen this, okay. That is not a muscle property, that is an active process. All these go into the system and displacement is produced, right. This is the actual movement. All these things control, and so the output from the CPGs are these, the output that is desirable or the desired output is this, right. And there are other things that control this. What are these other things? That is the sensory information that says whether you have reached the displacement that you want to reach and whether you have reached the velocity that you want to reach, right. There are other things that are not discussed, not shown in this picture. Please do check this. This is also responsible for swimming in other animals. For example, you could check this lamprey swimming. Okay. Important to note is the controversial role of CPGs in humans. Now, CPGs I have discussed so far are definitely have been demonstrated to exist in animals, definitely in lower mammals, higher mammals, but as you move higher up in the ladder, questions regarding its existence start to appear, especially in humans. What is special about humans? Well, the following is special in humans. We do not walk like a cat. What we do is bipedal locomotion, right. So, only 2 legs. Cats have 4 legs. So, fundamentally different, although they are mammals. So, little or some evidence for existence of pattern generators in humans have come forward, but still that continues to be controversial. So, so this cannot be extended from elsewhere, but at least the following is known in lower animals, especially in cats, in many other for example insects. So many hundreds and hundreds of papers have shown the existence of these pattern generators that are responsible for locomotory movements in lower animals and vertebrates, as in non-mammal vertebrates, etc. So, that is central pattern generators. Okay. So, what have we seen in today's class? We have seen spinal cord and injuries to the spinal cord, and injuries to the spinal cord, depending on where the injury is happening, this could cause hyperreflexia or hyporeflexia. If it is happening at the peripheral system, hyporeflexia. If it is happening in the central, it can be hyperreflexia. Then a specific form of hyperreflexia or hypertonia, specific form of hypertonia is spasticity. This is the rate dependent rigidity. When the stimulus or when the stretch is applied very fast, the rigidity is going to be great. When the same stretch is applied relatively slowly or very slowly, the rigidity is going to be smaller or even may not be felt, right. So, this is the special case of spasticity, and we have seen how central pattern generators can produce rhythmic activity. Okay. So, with this we come to the end of this class. Thank you.